Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, patient advocate Donna Cryer, liver wellness advocate Louise Campbell, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives. On the Surfing the NASH Tsunami podcast.
For everyone with an interest in NASH or more broadly fatty liver disease, Surf's Up, episode 36 of Surfing the NASH Tsunami. The second of our 3 daily AASLD summaries for 2020 starts now. This is the end of day 3 of the Digital Liver Meeting, or AASLD 2020. I began yesterday by discussing the excellent NAFLD Special Interest Group presentation Friday morning. Stephen and Manal were here then when I was talking about it, but not Mazen. Mazen, congratulations on a great opening presentation.
Thank you so much. It was a pleasure.
Thank you. And while we're sharing kudos, the last thing I got to see before I came here were the late breakers, including an excellent presentation that Naim did on the abstract that he'll be talking about this evening. Naim, that was also fantastic.
So much. Great to be here tonight.
Without going into the details, we're having some interesting technical challenges right here. So we can hear Louise, but we can't see Louise. Louise, say hi, just so we're sure you're here.
That's probably the best way to be.
Well, okay. At any rate, thanks for being with us. Today is going to sound a lot different from yesterday. It's going to sound different because, as expected, we have Mazen and Naim with us instead of Stephen and Manal. In addition, though, Donna Cryer came up with something she needed to attend to this evening. She won't be joining us either. Louise and I will attempt between us to present the paper on lean NAFLD that she was scheduled to present tonight. But since we're each roughly half prepared, that could go anywhere from 25% as well if it's multiplicative to 100% as well if it's additive. We'll find out when we get to the end of it. So we have a smaller crew tonight. We have no extra sode. It's been a long day for all of us. It's 1:45 in the morning for Louise because our technical got started a little bit later than we wanted. So I would just like us to jump in. Let's use the same icebreaker that we used yesterday, Louise. Our conference special, which is, what's the most important or exciting thing you've read or heard since this conference opened? Brave one, go first.
Well, I'm encouraged to see all the new treatments and therapeutic regimens showing success in 2020. This is very different than 2019. I was a little bit depressed in 2019. We had a few failed, even phase 3 programs, and nothing was looking good. And there was a lot of buzz what's going on with the NASH therapeutic landscape, and hopefully 2020 will put an end to this. We have very effective regimens. We have biopsy studies that are showing significant NASH resolution, fibrosis regression, and in some of them, they're hitting both endpoints, the fibrosis regression and NASH resolution. In terms of reduction in liver fat by MRI-PDFF, we are hitting very high marks now. So that 30% relative fat reduction is being achieved in 90% in some of the regimens. So this is very encouraging. Several, non-invasive tests from AST to ALT, cytokeratin 18. The FAST score is the new kid on the block, and it seems like it responds to treatment very nicely. So very encouraging, and I'm very happy to see that the NASH landscape is looking positive.
So Naim, not to give away what you're going to talk about on the podcast, but when you said 90% during your late breaker today, I swear my head did a 360 like a cartoon character.
This is 6 months of treatment, so this is really impressive.
Okay, thank you. Mazen, Louise, go ahead, either one of you.
Along the same line, I think the New England Journal published the SEMA on their webpage when we started the meeting, and that was big news. And Phil Newsom presented this great abstract with a significant NASH resolution, up to 59%. We have been talking about combo forever. We have written paper back in 2014 and 15 saying this is the future. It's like diabetes, and we're going to combine other medications to metformin. And here in NASH, Naim presented it, and as you said, it was 90%. I'm going to use abbreviations for all names since the names are getting complicated. Mm-hmm. So lanifibranor also had very impressive endpoints with both NASH resolution and fibrosis improvement. Not to underestimate diagnostics. So there were symptomatic data. We had, Naim and I and Steve had an abstract about the MAS score, which is MRI-based. Mm-hmm. We also had metabolomic-based tests to detect NAS more than 4 and F2 and higher. So all those are exciting. However, we still have more questions. Like the semi-exciting presentation earlier today did not explain why they didn't have a fibrosis improvement, although you can argue the placebo was quite high. So we have way more to do. So once we go back from this, we'll do more.
Yeah. Well, it's because there are always things to learn. Louise, go ahead.
And I'm going to go for one that I participated in today, and it was the late breaker, and I think it's Kathleen Alkhouri, and it was a study looking at NAFLD cardiovascular risk, and it was based on a PROMISE study in cardiovascular disease, where they happened to image 3,756 livers of 10,000 patients, and 959 of them had steatosis. But the startling piece of information that came out of that was that baseline hepatic steatosis was an associated risk in 70% increase of major adverse cardiovascular events. So it was astonishing in the level of cardiovascular disease-related events due to baseline steatosis in these patients. So it's emerging data, it's awaiting publication, but it was a very interesting Meet the Expert session.
So I did something that demonstrated either that my brain is truly bimodal or I'm halfway schizophrenic, but I listened to the late breaker and the COVID in the liver at 5:30 at the same time, each in one ear. And yeah, Mazen, I actually did that.
That worked?
Kind of. Multi-braining, but actually
multi-earing.
In some points I was only on one, in some points I was only on the other, but I had both in my ear at the same time. If I wanted, so I could take a headphone out. But I was struck by, in the COVID, not only the biobank, but also the work from Latin America, which again came back to the idea that liver is not merely an organ that is damaged by COVID, but in fact, liver, particularly combined with obesity, is a predictor of the severity of COVID infection in individuals, which when you put that together with the cardiovascular data, speaks more loudly to the importance of treating fatty liver disease. So it kind of feels to me that what's converging in this meeting at once: great drug results, a better understanding of diagnostics and the ability to use diagnostics both to evaluate patients and to learn about, and then an elevated sense of why the liver matters. We talk sometimes on podcasts about respect for the liver. I think on top of having better drugs and everything else, the liver is getting more respect this week, and that's an exciting thing in its own context.
Indeed.
Okay, so with that, why don't we go on? The rest of this program is fairly simple. One at a time, each surfer will share a 5-minute summary of poster presentation chosen to present today. I'm going to ask Louise to go first, A, because it's late there, and B, because Louise is going 1.5 or maybe even 2 times in the next hour. So, Louise, why don't you start with your original paper, and then we'll go forward from there.
My original paper was Oral Abstract 90, which was cirrhosis regression is associated with improved clinical outcomes in patients with non-alcoholic steatohepatitis. And that was a run Sanyal and the team, and again some great co-authors on that. And I always hate it when I look down and I see Stephen, so all I can say is I hope again I do this one justice. And this was a study that was basically looking at the clinical relevance of cirrhosis and regression. So cirrhosis is the primary determinant of adverse outcomes for NASH, which we know. Regulatory guidance for NASH cirrhosis encouraged event-based trials rather than histological endpoints. So what they did was look to evaluate the frequency of cirrhosis regression in NASH in 2 clinical trials. And they also wanted to evaluate the association between cirrhosis regression and changes in the non-invasive tests of fibrosis in patients with cirrhosis, and to evaluate associations between fibrosis assessed histologically and the non-invasive tests with liver-related outcomes and complications in patients with NASH cirrhosis. So they combined the data of 2 clinical trials, which mainly included similar inclusion criteria, if not almost identical. They were both terminated through a lack of efficacy. So simtuzumab and STELLA-4. And I think the key inclusion criteria that they used and combined was histologically confirmed NASH, compensated cirrhosis F4, Child-Pugh males of less than 12, platelets over 100, 10 to the 9, and STELLA-4. So they randomized, stratified by diabetes, portal gradient of greater than 10, an ELF of greater than 11.3, and all of these studies, as we know, were terminated due to lack of efficacy. And I think it's worthwhile saying that they were clear that the biopsy for these studies were done by the same histopathologist and centrally read. So what they wanted to do as their assessments and endpoints was study assessments and endpoints was NASH CRN for the histology, Ishak and modified NASH, morphometry for hepatic collagen, and alpha smooth muscle expression. So the non-invasive tests they were looking at were ELF, NAFL fibrosis score, FIB4, liver stiffness with FibroScan. So the cirrhosis-related clinical events that they were looking for were ascites, portal hypertension-related hemorrhage, hepatic encephalopathy, a MELD of greater than 15, or liver transplantation or death. So they looked at statistically, they wanted the associations with regression and clinical parameters, and they used a Fisher exact and Anakova. And for associations between histological findings and the non-invasive tests or clinical events, they used the Kaplan-Meier-Clock's regression. So they were assessing baseline and change from baseline and last observation carried out. So the study population overall were comparable, although the platelet counts in the SIM study were slightly lower than that of Stella 4, suggesting probably slightly more advanced disease. So going on to the results, liver-related events, median follow-up was around about 16.6 months for these patients, and 71 patients went on to have a liver-related event. Ascites, MELD of greater than 15, number of 5, death, only one patient died, portal hypertension-related hemorrhage was 11, and encephalopathy 20, with 34 on the ascites. Those with Ishak, it went on to show, had a greater number of events than stage 5, so stage 6, fibrosis over stage 5. So they also had higher hepatic collagen, and as well as a alpha smooth muscle from baseline change of more than 5%, which was statistically significant. But the impact of fibrosis on these events basically all increased the clinical events, and the higher the NIT at baseline correlated greatly with the rise and reduction in fibrosis as well. So it was corroborated by the non-invasive tests at baseline. Changes in baseline were either associated statistically significant or a trend in that direction. Changes in fibrosis was associated with events, and in relation to fibrosis regression, 176 based on the CRN NASH, and 300 patients based on Ishak fibrosis score actually showed a reduction and an improvement in their histology and regression. Now that for the NASH CRN has a hazard ratio of 0.16, and for the Ishak fibrosis it was at 0.08. Patient characteristics in that regression were predominantly female, and patients had a obviously lower Ishak score. Their platelets were better, and patients with cirrhosis regression had milder disease, was the conclusion from there. So the strengths and the limitations of the study, large sample size was a strong strength, centralized review of histopathology, consistent with the associations between non-invasive technologies, but the limitations with the small number of events, limited follow-up, and the absence of effective therapy. So ultimately, they concluded that patients with compensated cirrhosis due to NASH, if it regressed of cirrhosis, they observed 16% over 48 weeks. Cirrhosis regression was accompanied by a decrease in hepatic collagen and several of the non-invasive technologies. Regression of fibrosis was also associated with a substantial decrease in liver risk-related clinical events, and data to support the utility of histological fibrosis regression and non-invasive technology as endpoints in clinical trials for therapies of NASH cirrhosis. So I suppose it followed very much on from my theme yesterday about the use of non-interventional therapies to try and monitor people in real life, and I suppose this is just more evidence to that. So that was my session for the day.
Thanks, Louise. Questions, thoughts from either of you gents? Let me see you nodding thoughtfully.
I mean, I think it's important data because, at the end of the day, we all want to know what constitutes a meaningful response on these NITs. And I'm speaking specifically for the cirrhosis regression one. And obviously, changes in the Ishak fibrosis stage and collagen content in the liver and smooth muscle, staining. These are, expected to show the, improvement in cirrhosis, but also the ELF, NAFL fibrosis score, FIB4, and transient elastography. And now we kind of have a threshold to show us, the hazard ratio for improvement. So I think this is very important data for us as clinicians because this is, the future. We're going to see patients with NASH cirrhosis coming to our clinics. They're going to ask us what's going on. Am I improving? And to subject them to several liver biopsies is just not reasonable, especially with cirrhosis. I do believe that the risk of bleeding seeing is potentially higher. And I think we need these NITs to show us if we are making progress with the treatment regimen. Do we need to switch? Do we need to add another medication? Hopefully we'll have, a few to choose from. And this data is very welcomed and important.
I personally think it's breakthrough, along with what Naim said, but I want to focus on one point in particular, the regulators. So the regulators, let me look at this as highway, two ways. We were going forward and saying, or we're going in one direction and saying, well, ELF more than 9.3 at baseline predicts cirrhosis, more than 11.3 predicts events, increased stiffness predict cirrhosis, predict events, yada, yada. So what the regulators probably said, or you hear them here and there saying, well, I want you to show me that NITs or reversing NITs can lead to decreased events, similar to the histology or regression of cirrhosis. So I see this as an opportunity or a landmark study that NIT's improvement predicted better outcomes in correlation with histology. Now, what's missing from this presentation, which I heard that they're working on, is the area under the curve, MPV, PPV, and all that. But I think, and I hope, this will be a shift in the mentality, the way the regulators will look at things. Liver biopsy is great. This is a disease of 80 million people. This is a step to move away from the biopsy, which we'll use here and there, but this is important.
I think this is the last frontier when it comes to NITs. If you think about it, Roger, I mean, the context of use for these, number one is to identify patients with advanced disease. And I feel confident that we can identify patients with F3, F4 without needing a liver biopsy. The second thing is predicting prognosis. And I think we have enough data to show that these NITs can predict prognosis beyond biopsy. I think the missing piece is, can you predict response to treatment? And I think this will help answer that question.
Yeah, I had a similar reaction, by the way, Naim. Cirrhosis is in some ways the most interesting place to look, right? Because it's where you're kind of sitting on the edge of the razor, if you will. We're being able to identify marginal improvements will have the greatest short-term impact on mortality, morbidity, quality of life, the whole everything. So I looked at this paper and I said, if we're getting to the place where on the edge, we can start to talk about whether we're pulling people back off the edge or not. That's a significant thing to be able to achieve without a biopsy. I agree wholeheartedly. Mazen, I guess I'm trying to figure out, as important as the regulators are, I think if the tests are in practice and enough people use them, that over time, momentum will develop around them. So at that point, I try to figure out what. Now, I understand the idea that goes if the regulators get on board, it goes a lot faster.
I agree, Roger, but think about it this way. How many patients were in that study? Mm-hmm. We had about 2,000 people in that study. Wouldn't it be nicer not to biopsy 2,000 people? The first study did HVBG, and now all of us know how hard it is, especially there's the obesity component of the HVBG, which I'm not going to talk about, but wouldn't it be nicer just to design a study with confidence that NITs will take us where we like to be? And I think this is a very good first step.
Completely agree with you. And if Donna were with us tonight, she would make the point that having been through 6 biopsies in her life.
This study used Ishak, and a lot of the research that's been published throughout the conference uses Metavir. Now, obviously, Ishak, using its 6 differentials, allows you to look from Ishak 4 to 5, whereas in Metavir, would they not both be classified as purely 4? Therefore, you might not see that split that was shown in here.
I think Ishak is more granular and will show us more details, and I think it was smart to look at it this way. You want to look at the Metavir. The 4 equivalent in the Ishak is 5 and 6, and a lot of us want to see these changes between, like, the more granular changes from 6 to 5, 5 to 4. Even there's whole concept about collagen contact, thick septa, thin septa, to pick up any changes. So I think that was brilliant to look at it that way.
When I was looking at this, I was thinking about a paper that Path AI did at NASH Tech this past January, last time we were all together, many of us were all together, where in fact they used Ishak, and what they were able to show was that the pathologist coding wound up translating into Ishak scores that looked nothing like what you would have expected. There was one case, Thane, you nodding, maybe I'm remembering the same thing, where Medivir was getting constant scores of 4, and the Ishak actually wound up being 4.5 when you put it through the artificial intelligence routine, which in and of itself made me an Ishak fan immediately in that moment. A, because of the granularity, and B, because when you look at people estimating things, different phenomenon in some ways, but not. 90+% of all estimates out of 100% end in a 5 or a 0. Life doesn't work that way, but people tend to go to extreme values they're comfortable with. So when I saw all those Medivir 4s translating into an Ishak 4.5, part of my reaction was 4 is an easier place to think your way through than 3, 2, and 1. The more points you have on the scale, the harder it gets for people to default to a single value and say, gosh, that's a mess, which is really what 4 might be. So I thought using Ishak was brilliant. I think Ishak should be where we all look, frankly, going forward.
Or the path AI and other artificial intelligence technology, as you know, they break it down by one patient, and they have data showing you this patient is 60% F4 and 30%, for instance, F3 and all that. Those data are important, and it shows you how we should go deeper and beyond just F4 cirrhosis.
Yes. Yeah, in many ways. Couldn't agree more. Naim, I gave you such a build-up. Why don't you go next?
All right. Well, today I presented a late breaker from Gilead and Novo Nordisk on their combination trial. So I'm going to share some of that data with you guys and give you my personal interpretation. The safety and efficacy of combination therapy, including semaglutide, which is a GLP-1 agonist, cilofexor, an FXR agonist, and firsocostat, which inhibits de novo lipogenesis by inhibiting an enzyme called acetyl-CoA carboxylase. And all of these mechanisms of action play a role in NASH development and progression. So the aim of the study I presented was to evaluate the safety and efficacy of semaglutide monotherapy versus semaglutide in combination with firsocostat and cilofexor. The study was a 24-week study. There was no placebo arm, so all patients received active drugs. There was the semaglutide monotherapy, and then we had sema with firsocostat, sema with low-dose cilofexor 30 milligrams daily, sema with cilofexor high-dose 100 milligram daily, and then we had triple combination therapy. We escalated the dose for semaglutide from 0.24 milligrams gradually to 2.4 milligrams weekly, and we increased the dose every 4 weeks. And as I said, the primary endpoint was safety efficacy, but we also looked at, safety and tolerability, but we looked at efficacy endpoints, including reduction in liver fat, liver stiffness, and serum biomarkers. And we aim to include patients that have NASH and F2, F3 fibrosis, although we included many patients that didn't have a biopsy, so we identified this by MRI-PDFF more than 10%, liver stiffness more than 7%, and we did not want to include cirrhotics in this trial, so we used the Fibrosis test less than 0.75 to rule out the presence of cirrhosis. So when we looked at the patients we enrolled in this trial, 55% had diabetes, and there are some imbalances in regards to weight and ALT, AST values at baseline, but nothing that was statistically significant. And when we looked at the safety and adverse events, we had several adverse events, but they were mainly related to the known GI side effects of semaglutide, and grade 2 or higher adverse events were similar in the different regimens. We had 2 serious adverse events, one in semaglutide monotherapy, that was diarrhea and vomiting leading to dehydration, AKI, and then we had 1 episode of pancreatitis in the sema and high-dose cilofexor. No patients died in the study, and in terms of common adverse events, they're mainly GI. There was mild pruritus in patients with cilofexor. When we looked at lab abnormalities, we had a few grade 3, grade 4. We had 1 patient increased triglycerides more than 500 in a firsocostat arm, and 2 patients with cilofexor that had increase in CPK. It seems like semaglutide had a positive effect on the lipid panel, as we know, but it also mitigated the known negative effects of firsocostat on triglycerides and cilofexor on LDL cholesterol. Although it wasn't 100% complete mitigation, it seemed that it helped. All the patients lost weight, and I think this is related to the effect of semaglutide. There was slightly higher weight loss in the arms that contain cilofexor, and we've known this with other trials with the FXR agonist that there is a slight weight reduction. When we looked at MRI-PDFF reduction, we saw really impressive results. So when we looked at, absolute fat reduction, it was significantly higher in the combination arms, the ones that contained firsocostat, and this is consistent with the known effects of firsocostat on fat reduction. And then we looked at relative fat reduction, and we had different thresholds. But let's start with the more than 30% relative fat reduction, which has been consistently shown to predict histologic improvement. We reach higher than 90% reduction of liver fat by 30% or more in the combination arms. So this is really impressive. And then we, made the bar higher, and we went to 50% and 70%, and we showed that the combination arms were more effective than semaglutide monotherapy in achieving these higher thresholds. And it seems like liver fat reduction happened quickly by week 12 and continued throughout week 24. We also looked at biomarkers, and everything improved, and it seems like combination arms had slightly higher improvement in ALT, AST, CK18, potentially gamma GT. The ELF score increased in all the arms, and this was not different. And then we also looked at reduction in liver stiffness by transient elastography. There was reduction in all the arms. It wasn't significantly higher in combination. When we looked at achieving that 25% reduction in liver stiffness from baseline, this has been studied previously, and in the STELLAR program with Gilead, it's been shown to actually predict less disease progression. And it seems that higher percentage of patients on combination therapy achieved that 25% reduction in liver stiffness, up to 60% in some of the combination arms. And then we looked at the FAST score, and as I said earlier, this is the new kid on the block. And I really like the FAST score as a way to assess response to treatment, because you're hitting all the major players in NAFLD. So FAST has CAP as a reflection of steatosis, it has AST as a reflection of inflammation, and then you get your liver stiffness to assess for improvement in fibrosis. And the FAST score actually improved significantly more in the combination arms, and the highest improvement we saw was with the triple combinations, so semaglutide, firsocostat, and low-dose cilofexor. The FAST score decreased from baseline by almost 40%. And keep in mind that the FAST score goes from 0 to 1, so a reduction of 0.39 is a significant reduction in the FAST score. We're still learning what is a meaningful decrease in the FAST score, but I think this is compared to other data that we've seen at the meeting. This is a very impressive and robust reduction in the FAST score. Of course, we looked at glycemic parameters, and as I mentioned earlier, there's no placebo in this trial, so everyone received semaglutide, and there was a significant reduction in A1C as expected, but also we noticed improvement in glucose, insulin, and HOMA-IR. So the conclusion was that, again, the combination therapy with sema, silo, and firsocostat was well tolerated. The adverse events were mainly GI related to semaglutide. It seems that there was some mitigation in the increase in triglycerides and LDL, so that was reassuring. And in terms of efficacy, we saw improvement in serum biomarkers, liver steatosis as assessed by MRI-PDFF, but also CAP, and also liver stiffness and FAST score. So I think based on these results, this provides rationale to continue with this regimen, especially the triple combination therapy, and potentially the next steps will be to design a biopsy study where we look at improvement in histology with this regimen.
Powerful paper, very nicely presented. I have to admit that when I saw the SEMA monotherapy paper, I was curious about the lack of response in terms of fibrosis. Everything else made sense. We all know what SEMA is. It's a fantastic GLP-1. It's the best in that class when administered subcutaneously, but the lack of NASH. So when I started seeing the results that you were sharing today, I thought those are pretty, as I think I said, head-snapping, because they suggest that you can then take the benefits of SEMA, put in a combination therapy, as Mazen points out, something we've believed in forever, and get results that are far more, it does feel like the sum is far greater, the whole is far greater than the sum of the parts in this particular study.
And I think this was timely because we just published the results of the Atlas trial, and we looked at that combination, and that was a tough population, Roger. I mean, they're F3, F4, we had 56% with cirrhosis. And although, in the press release and even in the publication, we didn't achieve, that statistical significance, numerically, if you look at at the silo low dose and fursocostat combination, there was regression in liver fibrosis by one stage in 21%, and that was, compared to placebo, it was 11%. And again, as I said, mainly cirrhotic trial, I think that was impressive. So I think there is merit to, silo furso, and now you add sema, and you get the weight loss, you get the fat reduction, and you also get the cardiovascular benefits. I think this is powerful.
Roger, you nailed it, actually. That was exactly my impression. If you want to put the day together between early morning ending with Naim's presentation, with a significant NASH resolution on biopsy that we saw earlier, and these results that Naim presented, it makes complete sense that this combination should go ahead to make sure we get the fibrosis points. A couple of points I want to also add to that. It's some people asked about the placebo. We saw it. Naim answered that it's a proof of concept, but you cannot get that 9% with placebo. We know that for sure. Another point that caught my attention, I'm one of the investigators, is the titration. So we have to keep in mind that this dose was titrated to the optimal dose at around week 17. So you gave these people a very small window to show improvement. And you just think about it, if you go beyond, and this has been shown with the semi-weight loss programs, that you still have a lot of more room for improvement. So those are things that they strike me in addition to the significant reductions that we had in this study.
Yeah, the only thought I'd add to that, and then Louise, is that if you take a look at weight loss programs in general, right, success feeds on success. So the ability to work from a platform of SEMA, getting to the right dose, and then starting to drive some behavioral change on top of what we're talking about, it feels to me like everything should potentiate everything else.
One of the trials is looking at semaglutide, the same dose, 2.4 weekly, with intensive behavioral therapy. And I think that's the way to go, because hopefully at some point we will be able to de-escalate some of these drugs and maybe start with triple combination, but then end up with monotherapy, especially if they are able to maintain that weight loss.
Agreed. Louise, you have anything you want to add?
I think it's an exciting set of combinations, and I think it's the one thing that I've also seen diabetes and obesity societies very interested in. So there's an aligning of the planets between all the three areas there, which is good. And I think on behavioral change and lifestyle change, I think use of non-invasive technologies that patients can see and interact with is a very, very strong motivator. And I think that added has the potential to enhance all of the medications and potentially, as you say, reduce the doses to the best optimal dose for each individual. So, no, it's very exciting.
Yeah, I'll just make one more comment and then let's go on next paper. Back in my marketing research days, I actually was invited to advise Novo on the launch positioning for Lira. Before when GLPs were kind of like a glint in people's eyes. And you could see that this class had the potential to be true metabolic agents in a way that nothing else had been a true metabolic agent. To use an American sports metaphor, touched all the bases. And then the question was, how long was it going to take for that class to achieve its full potential? And I think we're starting to see that now as we bring liver into it, because metabolic liver is important, and we've got anti-fibrotic agents, and we can just do some remarkable things. Very exciting. And
they have cardiac data that is favorable, so it's coming together, the fibrosis, the metabolic, and the cardiac, so it's good.
Very, very good. Okay, Mazen, why don't you go ahead? Next paper.
So yeah, let me try to make sure Naim gets out to his dinner, so I will be timing myself. So this is the efferoxamine by Akero. Again, I'm going to go back to the abbreviation EFX, presented earlier today, and it's, as you know, it's FGF21, and we have the FGF19s and 21s. And as you know, they work on RTC, R2C receptor and RC3 receptors in the liver. different effects on de novo lipogenesis, insulin resistant, and the bile acid, which we can discuss another time. But what is this? The design is a randomized control trial, double-blinded placebo. The inclusion criteria included the following. They had NASH patients, biopsy proven with F1, F3. Of course, they had to meet the NAS more than 4 to be enrolled. Again, F1 to F3, and they had to have a PDFF more than 10%. So there were 80 patients in this trial, 20 each arm, placebo, 28 milligram, 50 milligram, and 70 milligram. And that was once weekly injection in this trial. So let me talk about the endpoints in general, and follow me about the design of the trial. So the primary outcome, this was a 16-week study. The primary outcome was at week 12, which is the relative reduction in PDFF, the 30% that we know of, as well as absolute reduction as a secondary endpoint. The caveat in this study that people who responded to PDFF at week 12 went ahead and had a liver biopsy at week 16. So that's important to think of. There was discontinuation, 11 patients discontinued for quote-unquote administrative reasons, 7 adverse events. So we ended up with, out of the 80, 68 in the PDFF analysis. Out of the 68, we had 50 responders. And the 50 responders were 48 on the treatment, EFX, and 2 on the placebo. Again, the PDF responders, 50 out of the 68. So these 50, then patients, if they responded, they had a liver biopsy at week 16. And the people that they had biopsy are only 42, 40 of them on the EFX, and 2 of them are in the placebo. So that's how the study went. Let me report the results to you. So characteristics, 2/3 had F2 to F3, so it was enriched with fibrosis, clinically significant fibrosis. 50% were diabetics, a little bit more on the placebo side, and those are the results. So the relative reduction on PDFF was as following: 0% on the placebo, and then you see dose-dependent response. 63 on 28, 71% on 50, and 72% on the 70 milligrams. The absolute reduction was 0, 12, 13, and 14. Similar to what Naim presented, people started looking at the more than 50% and more than 70%. So the more than 50% on the treatment ranged between 69 and almost 100%. More than 70% reduction on PDFF ranged between 50 and up to 80%. So those are also striking results. Normalization of fat on PDFF was also looked at, and the placebo was 5% in comparison to the treatments arm combined was up to 67%. Moving on to fibrosis markers and, ALT and AST, PRO-C3 dropped, ELF dropped in the treatment arm, not on the placebo. ALT dropped by 40% in week 8. And now I want to report to you the histology. So the histology actually was very impressive. They looked at both NASH resolution without fibrosis worsening, and the other way around, fibrosis improvement without worsening steatohepatitis. Again, I want to remind you that the people had biopsy, they had to be responder to PDFF. Right. So they included placebo because the ethical thing was to do biopsy in a short time if they responded. So the placebo, if they responded to PDFF, they actually had NASH resolution of 50%, but those were responders. A lot of them lost weight, or the 2 patients. The NASH resolution on the treatment was 48%. So keep that number, 48% on histology NASH resolution. The number for fibrosis was similar, was also 48%. So good histology response results. They also looked at 2-stage improvement of fibrosis. There were 11 patients that they responded. Let me take you to the side effects profile. The side effects profile, there is a signal with FGF, or this one, including GI side effects. There was nausea as well as vomiting, nausea up to 34%. There was increase in appetite. However, there was no change in BMI. Discontinuation rate was 1 patient on 2 patients on the medications. There was 1 patient on the 70 milligram that had acute pancreatitis. and had diabetic ketoacidosis. Let me finish with glucose and lipid panel. A1C improved up to 0.5 milligram, but if you looked at the diabetics, it improved actually up to 0.9. The dyslipidemia also improved. Triglyceride decreased up to 40% on the treatment. HDL increased up to 40% on the treatment, and non-HDL cholesterol decreased by up to 22%. So in summary, great PDFF results. Histology on responders of PDFF looked very promising in terms of NASH resolution and fibrosis improvement. I think what to do next is to see the data in a larger randomized controlled trial with biopsy, as regulator requires right now, without slicing that by response. But the FGF has proven either 19 or 21 to be a major player in the NASH game.
So I'm going to be talking about 19 in a couple of minutes, but I agree with you. I thought at the time, so one of the interesting things, right, these are studies that were initially published a few months ago, right, because timing in the world changed and everything.
Yeah.
What was there in this particular presentation that you thought was interesting that we might not have known back at the time of initial release?
I think the details that they went into in each one of them, the additions that they had compared to that time, I think the details of the 50% and 70%, it was quite striking. And the normalization of PDFF and the treatment arm versus the placebo. We had also the chance to look a little bit closer on the side effects. And if you look at different agents, this nausea and GI side effects is in GLP-1 as well. So this is one of the problems that we will face in the future. Mm-hmm. And also, the good news is the effect on the glucose and the dyslipidemia. So I think the more details that we got was very helpful.
Okay, great. Naim, question? Louise, question?
No, I mean, I think, the whole FGF21 class is becoming a very promising class in NAFLD. I think it didn't pan out the way we expected in type 2 diabetes, but, with the efficacy data that we are seeing now, I'm very impressed. I think not all FGF21s are created equal. So, it seems like the Akero compound is very powerful. It has to do with the downstreaming signaling with, whichever FGF21 you are using. We have several companies that have different FGF21, so we'll see who emerges as best in class. But I think, it's encouraging to see that potentially it has an effect on A1C, potentially it has an effect on weight. It seems like it was well tolerated. It's once a week. So I see it as, a promising class. I think I would like to see more safety data, look at effect on bone density, get that DEXA scan data out. But I'm very encouraged with what I saw. And, it's looking as potent as FGF19, and this is good for us.
Okay, good. Louise, questions, comments, thoughts?
My only comments are it's where you get a great result and you have to have a biopsy still, but it's like a win from that one, but I understand that it has to be done. And I think the other one is, in going back to the presentation that I discussed, if we can show that we can get resolution like that, we can show that we potentially get the improvement in all of the other areas. And we've now got ways to measure it, which is very, very exciting. So this year is a great year for development, and it sounds like it's going to continue.
So today and tomorrow, I'm going to be presenting different elements of the aldafermin work. I do that because tonight I'm going to talk about the results of the study. But there was a late breaker today that was not about the results of the study per se, but about the differences between F2 and F3, which I think are instructive. And there's a poster that we may get to tomorrow about the protocol that was used to manage increases in LDL that might result from aldafermin in terms of use of rosuvastatin and other things. And that result was at the end, ASCVD risk was decreased both for placebo and control groups using that protocol. So if I can get to that tomorrow, I will, but I certainly want to get to the late breaker tomorrow. Today, let's talk about the study. Okay, this was these are the final results of a 24-week cohort in the phase 2A aldafermin study. That study had 4 cohorts, 3 with different doses over a 12-week period, and then this one, which was the 1 milligram dose over a 24-week period. So this included a 1 milligram dose. The sample for cohort 4 included 53 patients receiving active medication subcutaneous daily. There were 25 receiving placebo subcutaneous daily. The inclusion criteria were biopsy-confirmed NASH with an NAS of 4 or greater, and at least 1 point each component, either F2 or F3 staging for fibrosis, using the NASH-CRN criteria, absolute liver fat content at or higher than 8%, and depending upon gender, an ALT of 19 or higher for women and 30 or higher for Okay. The non-invasive endpoint measures included PDFF to determine steatosis, ALT, AST scores and ratio for inflammation ballooning, ELF and Pro-C3 to take a look at fibrosis, and the biomarker C4 to assess bile acid levels. Primary endpoint was change in absolute percent liver fat. Liver histology, ALT, AST, and the biomarkers were all secondary endpoints. Aldafermin is characterized as causing increase in LDL-C in some patients. When that happened, rosuvastatin was started at week 2 if the patient had a 10-point rise in LDL, a 10 milligram rise in LDL or more. Baseline demographics are interesting. 9 of the 23 patients in the placebo group were male. The aldafermin group was split 50-50 between genders. That mattered because therefore you had different ALT scores in the two groups because the rule-out criteria for the different genders were different, and as a result, the group that had more men was going to have a higher ALT average score. Other than that, and a couple of other issues around that, there were no differences between the two groups at baseline. Aldafermin met its primary endpoint. It had a 0.7% reduction in absolute liver fat versus 2.7 for placebo, 39% reduction in relative liver fat versus 13. 68% of patients had a 5% or greater reduction in absolute liver fat versus 24 for placebo, and 66% had a greater than 30% reduction in relative LFC versus 29 for placebo. This study did not look at 50 or 70 or any of the higher numbers, at least not in what was presented today. Secondary endpoints suggested rapid, consistent improvement in fibrosis and NASH resolution by week 24. 38% had a one-stage improvement in fibrosis with no worsening of NASH versus 18 in placebo. 24 had resolution of NASH with no worsening in fibrosis versus 9 in placebo. What was interesting, and back when this was first reported, I think it's the first time I recall seeing this number, in a press release at least, was that 22% of patients exhibited both a one-stage improvement in fibrosis and resolution of NASH. And what makes that number striking is that number in the placebo group was 0. So fundamentally, all confirmed by sensitivity analyses. Among the F3 patients, 30% had fibrosis improvement of 1 level or greater versus 0 on placebo. 46 with the 30% improvement showed a one-stage fibrosis improvement, again, versus 0 on placebo. 62% demonstrated at least a 2-point NAS score improvement versus 9 on placebo. ALT, AST, Pro-C3 all achieved significant reductions by week 4, maintained them throughout the 24-week period. C4 dropped more than 60% versus a 1% increase in placebo. Serum bile acids dropped by about 55% versus a 30+% increase in placebo. Okay, no changes in blood pressure, heart rate, well-tolerated adverse events, generally comparable to placebo. The only adverse event leading discontinuation was actually in the placebo group. And one last look, if you compared the 1 milligram cohort that went out at 12 weeks to this 1 milligram cohort at 24, the 1 milligram yielded a 25% fibrosis improvement at 12 weeks versus 38% at 24. And this group, the 12% response was 25% in the 1 milligram group versus 42 in the 3 milligram group, which suggests that you could take the dose up, take the higher dose at the longer period of time, and possibly even get a better result. So in conclusion, primary endpoint was met, clinically significant improvements in all the histology, regulatory endpoints, fibrosis improvement, resolution of NASH, and that 22-0 score on the composite endpoint, which is striking. Cohort 4 showed that the 12-week effects were durable to week 24. And the tolerability profile appeared well tolerated and safe up to week 24. Did not have a 50, did not have a 70, did not have a 90, but it appears that it's a drug that will be well tolerated and will work well and do the things that we're looking for right now. So that's the paper in a nutshell. I couldn't have talked much faster than that, Naim. I'm really trying to help you with dinner. But so let's questions, comments, thoughts.
No, I mean, I think, in terms of efficacy, FGF19 has proven itself to be an effective agent. I think the higher the dose, probably you're going to see higher efficacy, but it comes with the price of more potential for adverse events. We always have to be mindful that patients with NASH die more from cardiovascular disease than liver disease, as far as we know, although there's some new data suggesting that liver disease may may contribute more significantly to mortality, but I do believe that cardiovascular disease is still a very important cause of mortality. I think, one point that I heard a couple of times now at meetings is the fact that we have to use statins, and are statins doing all the heavy lifting, and are they the reason why we're seeing improvement in histology and liver fat? And I don't think so, because 50-55% of my patients in clinic have type 2 diabetes. The majority are on statins, and they progress and they end up with NASH cirrhosis. So I think statins may have a synergistic effect with FGF19, but I don't think adding that statin is what's doing the heavy lifting. I do believe it's the FGF19. I think the pathway makes a lot of sense. It's complementary to FXR agonists. So I think this is going to be a regimen that will stay with us. It could be something that we talk about is this induction therapy, and maybe something that we use to induce a quick and robust response, and then maybe we can de-escalate the FGF19 and transition to a more tolerated drug without an negative effect on LDL. But I do believe that this is a promising regimen and very effective one.
Okay, thanks. Two things real quickly. Number one, in the watch this space category, we're going to come back to that issue of liver disease and CVD and what does NAFLD actually cause. At the end of the month, Ian Rowe from Leeds is going to come back on. He's done a rather interesting analysis or reanalysis of some of the work that suggests that it's about liver disease and got to a different finding, and we'll share that probably at the end of November, beginning of December. Second, one of the interesting things about statins that they reported, I don't remember if it was in this paper or in the NEF paper that I'll be talking about tomorrow, was that 61% of the patients coming into the trial should have been on statins based on diabetes and lipid level, but only 32% were. Mm-hmm. So one of the benefits of being more thoughtful about the use of statins in the context of NASH is we might actually get statins to more people who should have been having them in the first place.
Statins are underutilized in patients with NAFLD. So I agree. Maybe this way we can get more people on beneficial statins that they actually need regardless of the NASH treatment regimen.
You're talking to a guy who had to arm wrestle his wife for 2 years to get her to accept the idea that I should have been on rosuvastatin, which I am now, and it took my cholesterol down through the floor to where it belongs. So I'm appreciative of this argument.
Yeah, so you reminded me of another success story today, which is the NASH resolution and fibrosis improvement. You presented it in this trial, but we also saw it earlier today with lanifibranor, that they achieved that as well. I mean, they used the SAF score, but on the 800 milligram was 21%, and the 1200 milligram was 31%, I think, versus the placebo was 7%. I hope now they're not going to say, well, we should use this as achievable. No, it's a way harder endpoint. But I think also medication started showing good effects in this area. So that's a highlight of the day as well.
Agreed. So, Louise, thoughts about this, and then we'll jump on to the last paper.
No, I think the guys have said it all really well. And again, it's just another product that's showing great efficacy, I suppose. So, no further comment from me.
I'm mindful of all the people who were hanging crepe when Intercept received its CRL. And if we had all said that this was how excited we'd be in 6 months, I think folks would have been really surprised. We came up with an episode 3 weeks after that talking about what exciting things were developing, mostly because we just wanted people to calm down a little bit, but and not be so sad. But yeah, it's a very, very exciting few months and a lot of great stuff at this conference. Louise?
Yeah, I think it's exciting now, but there are still patients who were affected by that decision. And will be until these drugs come out. And I think
Absolutely.
We can't lose sight of that.
No, hope is pivotal. And there were patients, look, you're talking to somebody who had a relapse of melanoma that was not found for 3 months after it showed up on an X-ray. And in the intervening 3 months, Yervoy was approved. And I'm not saying that Yervoy was what did it. In fact, that's a longer story for another day. It wasn't. But knowing that Yervoy was close and knowing that Yervoy was there on the day that they told me that I had relapsed made a world of difference. For those who know the calendar on this, that was 2011. I'm still here. And I've been totally clean since 2011. So I appreciate the value of hope more than most people I know because I've got significant firsthand experience. I totally agree with your point. That's the tough part of the story. But looking forward, we have much to be hopeful about, more than I think people expected 6 months ago. And that's the other side. Why don't we go on to the last paper, the Ahmed paper? this was the one that Donna had prepared, and Louise and I have each taken pieces of. And Louise, why don't you get us started, and then I will fill in where I can.
Well, I was in the session, but it's one of those ones that I was listening to, but not really taking the amount of notes because I wasn't expected to do this. So it was abstract number 86, which was the natural history of lean non-alcoholic fatty liver disease, a longitudinal US population study. And it was presented by Omar Ahmed and the team from the Mayo Clinic. So for the background, they were looking at the natural history of lean non-alcoholic fatty liver disease being not basically well understood. And therefore, we can't counsel patients for disease management. It's poorly located, and we were looking at the US population particularly. So what they did was look at all adults diagnosed with NAFLD in Olmsted County between 1996 and 2016. And the ones that they identified then all went on to have subsequent medical events ascertained using the medical record linkage system. So they split them into three groups: lean, overweight, and obese. And what they did was they used the normal cutoffs that we have for Caucasians. So that was less than 25, 25 to 29, and 30 and above for obese. So they then looked at the probability to develop cirrhosis, decompensation, malignancies, cardiovascular events, or death among the three groups, estimated using the Allen-Johansson method, treating death as a competing risk. So the BMI categories of these outcomes were then explored using Cox proportional hazard regression analysis. So looking on to the results of that, basically 414 patients were identified out of 4,834 as having lean NAFLD.
Okay, 414 had lean NAFLD, 1189 were overweight, 3,231 were obese. Followed up for cirrhosis, CV events, malignancies, and death. As you pointed out, death was a competing factor in the analysis.
Yeah, so the lean was 414, as you say, overweight 1,189, and obese 3,231. So they did a 23-year follow-up on these patients, looking for cirrhosis, cardiovascular events, malignancies, and death, as I say. So when they looked into it further, the development of cirrhosis in the lean reference to obese, the hazard ratio was 0.3. So there really wasn't anything in the development of obesity, development of the NASH in the lean population. So it was obesity, overweight, and then the lean. So there wasn't any identification of problem there. Cardiovascular and malignancy, again, were very, very similar. There wasn't a particular increase in there. But when they went to mortality, there was a difference in the lean reference to those of obese, the hazard ratio of 1.63, and it was statistically significant. So what they looked at overall was that 7% of the population in the American is got lean NAFLD. They tend to have a lower metabolic risk profile because the patients had diabetes 20% in comparison to 40% in obesity against lean NAFLD. Hypertension, they had less of that, 30% to 45%, and dyslipidemia, 49 to 60%, and they were more likely to be women. So they had a lower metabolic risk profile. There was no evidence of worse liver-related outcomes, but they did probably have a higher mortality rate overall. So that needed an investigation a little bit more. And I think you also commented on that session from yesterday. So there does seem to be a reason to identify lean NAFLD because they probably have a higher mortality rate than some of the other NAFLD strains. But I thought it was a good presentation.
The key point really was the striking increase in early mortality, not total mortality. The mortality curve caught up over time, but mortality came earlier, significantly earlier in the lean NAFLD group. What fascinates me about that is really economic, which is that the expense in treating NASH really starts at F4, right? F4 is cancer, F4 is transplantation. F4 for people who cannot get a liver is a long, slow, painful, expensive fade. So if we can identify patients who are at risk of getting to F4 fast track, forget whether Mazen, it was you or Naim who used the number 3 or 4 decades on this podcast. I think it was on the podcast. Could have been one of the later presentations I saw during the day. It all runs together after a while. But if we can identify people who've got, say, a 10 to 15% risk of being in F4, serious F4, in 3 to 5 years instead of 35 years, then we can start to save money. And we can improve the quality of life of those individuals who are also the ones who are least likely to know that they're in trouble. As you point out, they've got lower levels of diabetes, lower levels of hypertension. They're not obese. They feel they're fine, and they're cruising along. I was at an investor conference, I think a Roth investor conference a couple of years ago, where one of the key speakers was someone whose mother went from feeling fine to being dead in 10 weeks. Perfectly lean woman, had no reason to believe she had a problem, lean, NAFLD, and they just, obviously, they couldn't get a liver quickly enough. So on top of everything else, there are some economics around lean NAFLD that say that if we can identify it, we can save lives and secondarily save money, but save lives and help people who have no reason to know they're heading into trouble. And I think that's the theme that emerges, I think, from the majority of this stuff. Not all of it. There's some that say that it doesn't work that way. But most of what we're looking at in this conference, I think, addresses that. Mazen, you have a funny look on your face.
I find your point actually really interesting, and I'm in deep thoughts where we should take these data. And you triggered my thinking that I totally agree the way you're thinking about this, but also I started thinking about screening. So as you know now in the United States, there is no recommendation to screen for non-alcoholic fatty liver disease. Once you find it, you stage it. That's what it says. But high-risk group, not necessarily the obese, the diabetics and others, they're not recommended to screen. As you know, versus Europe, EASL guidelines recommends that. And that was based on cost-effective analysis that older one that showed if you screen with a liver biopsy, it's not going to be cost-effective. Well, sure, it's not going to be. And I think Stephen Harrison did shout out to our paper, actually, that we published in Gastro this year, that we showed that screening non-alcoholic fatty liver disease patients is cost-effective in this country. Yeah. And it's that diabetic population that we studied, and it was cost-effective if you use the FibroScan. So to your point, we should start identifying these patients, and including the lean with diabetes, because they're going to cause another health economical problem, yada, yada. More importantly, also the patient themselves. Mm-hmm. You don't want them to drop dead like the one you just mentioned. No. You want to stratify them. They should not die early for reasons that we can avoid. Anyways, that was my thought. That's why that had a look on my face. The other point is, I think in that study also, for some reason they had less regression to cirrhosis. And I didn't follow that point during the presentation, but I wonder because of their dying earlier, that's why they're not getting to cirrhosis. I need to see the manuscript in a full paper.
Yeah, I had the same
great study.
I had the same people. Yeah, that one cirrhosis number statement did kind of jump out at me. So Louise is renowned among the podcast for her perspective that we should just screen everybody.
Well, I'm going to come on to that point because there was actually a very interesting abstract in that session, abstract 88, which was changing lipid markers throughout early adulthood is associated with the development of NAFLD in midlife. And it's the CARDIA study. And it is worth listening to this session when it comes out because it was startling how these children over the 25 years that they followed them developed and screening early. And I think looking at what we've been discussing in this episode may well be the one. If we can turn around these cardiovascular diseases, if we can prevent people from being ill, then we can become a health service rather than an illness service. So yes, I think we can stop people now we have technology to keep liver healthy rather than. And Naim, I know, likes that idea.
Yes, I know he does. I think we all do, actually. I haven't yet encountered anybody anywhere near this podcast that doesn't like that idea. So with that, given that we are now 2 in the morning where Louise is, and and then to the end of the hour and change that we set aside for this stuff. Why don't we roll on to the end? Last question for both of you. Mazen, Louise, brave one, go first. What is the one most important thing you think is going to come out of this conference based on what we've seen so far and what you know is coming?
Louise, go ahead. Ladies.
I'm trying to think. I'm going to have to come up with a different one from yesterday, but I do think looking at all of the data today, looking at where we're heading, is that we are coordinating the circle round from liver disease to cardiovascular to diabetes to now coming back to drugs that can target all of those areas in one go. And I think, or in combination, I think that is exciting. And I think that taking that forward to be able to look at preventative health and screening in the highest risk populations to start with and then further afield. But I think all of that is exciting and it seems to be coming together.
I will start on the same thing. It's coming together. It is the meaning that the glass is half full. I think in the last few years, we have done mistakes, not intentionally. We're just learning. We're just deciding which endpoint should be the best. There were endpoints that were implemented. Some made sense, some did not make sense. Now we're achieving those. Now we know there are people that are like drugs that they're going to win, and we should start thinking about combining them. And I hope in the next year or two, we'll start seeing trial designed for drug approval, moving away from liver biopsy as a primary endpoint. So I think this started this meeting, and I will see more coming.
I mean, I think, the efficacy of different regimens is very refreshing, as I mentioned earlier, but also I'm hoping that we will start to say goodbye to liver biopsy after this meeting. And I know I'm being provocative. I know my pathologist friends will be upset with me, but I think we have to be realistic and realize that we just cannot biopsy all these patients to risk stratify and assess for response to treatment. And we are realizing, every is just limited, and it has many issues even in clinical trials where we are using the best pathologists in the world. So you have to understand that the community pathologists are not well versed in all the different aspects and nuances of how to interpret a NASH biopsy. So I hope 2020, if anything good is going to come out of it, is that we will have more effective treatments and NITs and move on beyond biopsy.
So, Naim, as you leave, I have to tell you I'm disappointed that Donna was unable to be here today, because of that comment, she would have tried to figure out if there's a way to kiss you through the screen. This is such an important point for her.
Thank you.
And Louise and I would have paid good money to see that, whether it failed or succeeded. So thanks so much for being with us today.
Maybe she'll reach out after she hears the session.
Okay, that sounds fine. Okay, thank you very much. Okay, now let's go back now with that. Thanks, Naim.
No comment.
Okay, good points all. To me, the two, I'm going to go back to Intercept again. The two really bad things about that CRL were, first of all, patients with hope who felt dashed. Second, diseases tend to develop awareness in primary care and throughout the community when the first drug shows up. Because historically, the first drug has been when people start spending money on education. So for OCA not to get approved meant that education wasn't going to happen. What is happening instead, though, is a holistic consensus about the disease, not just what is a drug to use, but the idea of what is the totality of what drugs have to do, and really a better sense of how to diagnose and how to conceptualize what the disease is. So normally this is a top-down thing that gets pushed by the drug company with money. Here it feels like it's more of a bottom-up thing, kind of bubbling up through the research and through the energy of people who've been doing this for 20 years. But you can certainly feel which way the field is headed right now. And I think there's a clarity about it and a pointedness about it that will serve us well in research. And frankly, when drugs start to show up and when we get a couple more of the diagnostics on the stage, will help patients relatively quickly get the help that they've deserved for far too long and haven't gotten yet. And that's not how it usually works, but I think it's how it's going to work here. And I think that would be a fantastic thing for the patients and society. And frankly, look, I've been doing this with you folks for 2 years, but you guys have been in the fields for decades. And for those who've been tilling these fields for decades, that would be a fantastic end result for all that work. So
fingers crossed.
Yeah, fingers crossed sounds right.
When you were both talking there is the fact that I've said previously that my career has been around nursing and looking after people who died young, and liver disease is a young death. Actually, what can come out of this conference is medications and treatments that mean that they don't die as young. And I think that is always a great outcome.
That is a brilliant note on which to end, Louise. So thank you. Mazen, thank you. Naim, thank you. Louise, thank you for hanging in until 2 o'clock in the morning and actually presenting a paper you weren't planning to present at 1:50 in the morning your time. I think you definitely get 2 gold stars for that one alone. Okay. I want to grant another extra special thanks to our engineer, Mike Wilson, who took until 4 o'clock Memphis time this morning to pull last night's episode together. And I told him this would be straight, clean, and easy, but then it took us 15 minutes to get up on screen, and people had to leave early. So, Mike, I'm sorry. There's another bad night for you, I fear. I see a bottle of Pappy Van Winkle or something like that in your future, courtesy of me. So thanks for doing that. Thanks for Palatale Lee for going above and beyond, getting all these sound checks coordinated, to Buzzsprout for seamless posting and everything they do, and Eric Rounds, who's always fantastic. Thanks, as always, to you folks, our subscribers, our listeners who've made this leading fatty liver disease podcast in the world. You surprised me. In the week before EASL, we put up a pretty good episode and nobody listened because they were waiting for EASL. We put up Jorn's episode at the beginning of this week on why NASH drugs fail, and it's been the most successful first 5 days of any episode since the end of EASL, so you guys are obviously paying attention. Thank you for that. We will be back tomorrow night. Stephen will be here tomorrow. Mazen will not, Naim will not, but Stephen will be here. Dr. Mary Ranella will be here with us. Hopefully, Donna will be able to make her way back as well. She wasn't sure exactly how long the thing she was working on was going to take, but we're hoping it'll end. So, another day of AASLD in the books. Tomorrow, more papers, one more late breaker. Everybody enjoy. Everybody come back to listen to us again. Stay safe, stay healthy, surf on. We'll see you on the podcast. Good night.
Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, patient advocate Donna Cryer, liver wellness advocate Louise Campbell, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on the Surfing the NASH Tsunami podcast.